Okay, good afternoon. I think we'll go ahead and get started. So if you haven't seen my email yet, so I have already extended deadline for writing assignment 3 until Monday at 11:59 p.m. So hopefully this will give you a breather and give you some time to polish your writing assignment and add more of what needs to be added. And if you still submit by today's deadline, the original deadline, you'll be getting 10 extra credit points, which is equal to 1% of the overall grade. So it's up to you what you want to do. Okay. So hopefully that you will take this time to refine your writing assignment, also use feedback that you guys got from your TAs on your writing assignment 1 and writing assignment 2. Any general questions before I get to? I have a couple of announcements, but are there any questions, any problems, any issues? Yeah. So a couple of things. One is about the group work. I got a complaint from one of the groups, and I thought to share that with you. In group work, we're expecting that folks would work together and cooperate to achieve their goal. Do you have to like your group mates? Not necessarily, right? In real life, you'll have to deal with people that you don't really like, right? Sometimes you have to take a professor that you hate, or you have to work with somebody that you don't like, right? This is just what life is about, right? So, however, you have to work with them to get the work done, right? Because if you graduate and you go to a company and you hate your group mates or whatever, and some of them are really like mean folks, right? But you still have to work with them, right? Just to show some compromise, get the work done. It's not about them, right? You're not going to propose to them or marry them, right? It's about the work. So just try to get it done in the most efficient way and in a respectful way. So no matter what, so some groups will have maybe 4 males and 1 female, or vice versa. So they might feel excluded sometimes. Sometimes people get excluded because of their whatever the perceived identity they have. So that specific complaint I got was about one of the students. She was excluded for obvious reasons. So, but this is not really acceptable. And no matter what that person is, just treat them with respect, right? And just do your best. So, and leave your judgment somewhere else. Don't bring them to the lab. Okay, so um, so far we have looked at the phenotype. So you have successfully compared the wild type and the mutant phenotypes that we've given you at the beginning of the semester. So you have successfully identified how the mutant phenotype is different from the wild type. And then you wanted to fully characterize this mutant, right? So you wanted to know whether that mutant phenotype is dominant or recessive. And for that, you did WTM1 and WTM2. And that told you that your mutation is recessive, and also it's not sex-linked, right? Unless you have CINCH, I believe. So, but then you also wanted to know where on the chromosome is my gene located. So for that, you did mapping crosses. We crossed different flies, and based on the recombination frequency, we determined how far your gene is from other genes. And you came up with a chromosomal map, right? You constructed a map that shows how far your gene is from other genes. And in real life, I think in previous semesters, these mapping process students used to do these actual crosses in the lab, right? So you have 2 flies, they differ in a couple of traits. You cross them, pick the F1, back cross it to the dead. So these were actual crosses, and students would have to score about 1,000 or 1,200 flies from each cross. So this time it's easier, right? We changed the course a little bit. So now that we know where your gene is located as compared to other genes, we want to determine like the exact type of mutation, right? Now that we see that these are 2 different phenotypes, we want to know how these 2 mutations are different at the molecular level. Is there a deletion, insertion, substitution? So for that, we've agreed that we need to do the DNA extraction, and we have you guys have successfully extracted DNA from the wild type and the mutant. And then we kind of realized that we're not really interested in the entire genomic DNA. We don't really care about the 13,000 genes in Drosophila. We only care about one single gene, which is your gene, right? Ebony, black, vestigial, or cinch. So then we decided to do PCR. And we have done PCR using primers, specific primers, that would amplify both the wild type and the mutant, right? So we did PCR to amplify these two alleles, and we got amplification. And then we ran that on agarose gel. We did gel electrophoresis, and which showed some bands for some of you. Some of you didn't get any bands, and that's okay. So two things in here. So gel electrophoresis or PCR, you shouldn't feel any like bad that you didn't get results. If you didn't get results, it happens all the time, a lot of times, right? And when I was doing my master's, I ran about at least a few hundred gels, right? And PCRs, like numerous. And many of them wouldn't work, and I have to repeat them. And there's generally certain reasons. Sometimes you didn't add a specific reagent, you missed something, you had the wrong temperature, you did something wrong, right? And you have to troubleshoot and figure what that is. So it's not uncommon, right? So it's common that PCR as a technique works, right? We know it works, but sometimes just something happens, it just doesn't work, and you will never realize what the problem was, right? So, but people will try maybe different concentrations of things, different DNA concentration, different primers, different enzyme concentrations until you optimize your PCR, right? But once it works, it should work, but still you could get here and there, it could also not work for some other reason. So, okay, what do we do if your PCR didn't work? Well, one, we have an option on, I think, Thursday. We'll make this upcoming Thursday an open lab, so where you can come and redo your PCR or gel electrophoresis. But just to make this clear, this is an optional day. You don't have to come. So this is just if you really feel terribly bad about the experiment that didn't work, you can come and repeat it, right? But you don't have to come and repeat it. If your experiment didn't work or your gel picture doesn't look okay, you can use the figure from the prep TA's samples that they provided you. Or if that also didn't work, your TA will provide you with a picture of a gel that you can analyze and write about. Okay, so if you don't have like the gel results, if you don't have a good picture, we'll provide you a picture, that's for sure. If you want to come to the lab and do your own gel, that's optional, and there will be no extra credit. You don't have to do it. So, but it's an option out there that you want to redo the experiment, right? If you do it, there is no extra credit. If you don't do it, there's no penalty. That's only for your own learning if you want. So if you want to do that, reach out to your TA and let them know that you're interested in coming on Thursday, right? And we'll find this. I think we'll make it an open day. Maybe we'll have a couple of spots available during the day where you can come and do the experiment. Okay. So the gels, assuming that we now have two bands, so I have two gels, at least one gel that shows two bands. So even if your gel didn't work, at least some other groups gels worked, right? And you saw bands. Did everyone see bands in the lab? Even if it's not on their own gel, but everyone saw the bands, how they looked like? No? Oh, really? Whose lab are you in?
Richard's
Richard.
lab.
Okay. Okay. Gel, DNA gel, electrophoresis.
We saw it in the presentation, but not in the lab itself.
Okay, in the lab, nothing, it was just black.
Yes, we saw no bands.
Okay.
He believes that the students who created the gels previously didn't add like something I forgot like the red.
Oh, methyl red dye.
Yeah,
Okay, that's
he didn't add it. Didn't add that. That's fine.
okay. That's a good observation. So because generally gels, that's why you put a ladder, right? So this is a ladder right here. So this is a ladder that we know it must show some bands, right? So if your gel didn't show anything at all, I would believe that they probably didn't put gel red, the dye, right? So, um, but this is how you have like this is the ladder, like a marker that shows different bands of different sizes. So these bands, each one has a specific size that we know how big it is, right? So if this is maybe 1500, then I would know that these are also about maybe 1500, right? So, so if you still have the remaining PCR product from last week, if your tube of 25 microliters, if it still has product, you can run it again. Do you have left PCR product? No, that's fine. You can redo the PCR, or we can give you PCR samples that we know work, and you can just do the running again. So I highly recommend, if you have not seen anything, I recommend that you do it. Of course, if you have time. It doesn't have to be the entire group. If your entire group is not available, maybe a couple of folks. I bet not everyone would be interested in coming again.
What's the time?
We'll set that with the TAs. Your TA will let you know exactly, but it's going to be on Thursday. Yes, sir. You have a question? What band showed up?
Like,
Yeah, that's the ladder, which is like one part that shows. Yeah, if you have nothing, it's kind of hard to interpret, and something went wrong. So, but there's one thing: you didn't get any bands at all, right? So that means something is wrong with the entire reaction, because we're expecting to see at least a positive control that will show no matter what, right? We have a band that must show up, which was the GAPDH primers, which it's a housekeeping G, meaning like when we test this, we must see a band. And if we don't see a band, then that entire reaction didn't work. I don't have to. So that positive control is like when somebody takes a pregnancy test, there's always one red line, right? This is the control. And then you look at the other line that shows whether the thing was pregnant or not. So if you don't see that red line, right, it's like not seeing that positive control on the gel, meaning there's something wrong. Can't say that the sample doesn't have the G because that sheet didn't show it, right? We say that entire reaction just didn't work. Good point. So, assuming that you saw the gels would look something like this. You'd have the wild type would have maybe something like this, and the mutant will have something like this, or vice versa. So you'll have the wild type and the mutant. They could be of two different sizes, or they could be of the same size, right? So those are the possible options. You could have two bands that are of the same size. One is bigger, one is smaller. Right, so you have maybe the wild type is bigger than the mutant, or it's smaller than the mutant, or they could be of the same size. So our goal test for this upcoming week is to look at the sequences of these two bands and align them and go base by base and compare and see exactly where the problem is or where the mutation is. So from this week, assuming that we have two bands, if they're of different sizes, we need to figure out what is different, which one is bigger, right? If the mutant is smaller, that means there is a deletion, right? The mutant is deleted. And we need to figure out what piece is deleted, right? And if the mutant is bigger than the wild type, well, that means that there is an insertion. And also we need to figure out what the sequence of that insertion is. And if both the mutant and the wild type, they are of the same size, well, there must be a point mutation, yeah, or substitution, right? So that's the goal of this upcoming week. And I'll walk you through the process of the analysis today. But the main point is not to have you follow the steps, because the document will be on Canvas and you can just follow the steps. I want you to understand what is happening and what we're doing and why, right? Because I'm not turning into technicians, right? With all due respect to technicians, but we want you to be able to kind of think about what is happening and what and why we're doing. So what the plan is, we want to look at these two sequences, the wild type sequence and the mutant sequence, compare them and see what the DNA sequence difference is, and if that difference would translate to a protein difference, right? If that molecular difference would lead to a change in the protein or not. And then look at the actual protein, how it looks like. Okay, so this week, so let me open. Why am I not able to see Teams? Okay. It's kind of hard to find the team. Okay. So this document will be up on Canvas. And this is the pre-lab that you guys will do for this week. So, so in the lab, now that I ran these these gels, what I would do if I wanted to sequence those, I would go and look at that gel and look at that band and cut it using a cutter. Just cut that band, cut it out, and then put it in a little tube with buffer, so I can so that DNA in that band can be released into the solution, right? So once we have that DNA in two tubes, in that tube we can send it to any company or facility that does DNA sequencing, and they will sequence it for us, right? So the way this happens in real life is that we took those bands and cut them out from that gel, put them in a tube, and then send them to be sequenced, okay? And we'll give them primers, the forward primer and the reverse primer that we have used in the amplification, right? So if you want to sequence something, you will have to do a PCR of that specific sequence, and then take that entire band, you cut it out of the gel, and send it to be sequenced. So what we want to do is we have 4 tasks this upcoming week. One is to find the identity of the wild type amplicon. So when we did PCR on the gel, you saw a band for the wild type, right? There's a specific band, maybe of a certain size, 1500, 1200. We want to know what the sequence of that band is. We want to know the sequence, right? And then we want to compare it to the mutant sequence. So I want to compare both the wild type and the mutant. So we'll find the wild type amplicon first, and then we will compare it to the wild type. Then we'll see if the proteins have been affected, if the coding sequence has been affected. And lastly, we'll look at the protein structure, the 3D structure of the protein, how it looks like. So first things first. So let's go to finding the amplicon, the sequence of the band, the wild type band. So let's say we're working with, let's go to lipase to get the wild type sequence. And I'll be using ebony, but the same applies for all other mutations, except vestigial. It's simpler. So we'll talk about ebony, and in lab, your TAs will help you with the rest of the mutants as well. So, and just remember, when you do your analysis, the pre-lab work, which is due general on Monday, we're not checking it for correctness, but for completion, right? Just do your best. Just show some effort. So before coming to the lab, we know that you don't really know that stuff, and you're just trying, right? So we know we take that into consideration. So imagine that my gene was ebony. So I will type ebony, click ebony, and this is my ebony gene. And if I click over here, let me just make it a little bigger. I click over here, it says get decorated FASTA file. And I look over here and I see some sequences, DNA sequences. So what is this? What is that sequence? Is it part of the gene, the full gene, the coding sequence? What is this? So this is the full gene, right? This is the entire gene from the beginning till the end. So this is the full gene, including promoter region, coding sequences, 5' UTR, untranslated regions, 3' UTR, introns, exons, everything. Right, so you got the entire thing. Okay, so that's the entire gene. So let's copy this. Let's take this down. Okay, but the format is a little weird. Those I can just search Control H and replace caret E with just replace R. So it's caret, not 6. Replace R. Kind of get it, that got rid of the kind of strange format that we have. So you don't have to worry about writing these steps because they're all explained right here in the document. It kind of shows you exactly what to do right here, right? And I'm also recording this. So now that we have the format, we have the genes. Let's see how many letters are here. 7,339 letters, right, or bases. Okay. So when we downloaded this, it says we downloaded ebony, right? Ebony. Is this the wild type or is this the mutant? You think it's the mutant, right? And I think the vast majority of you would think that this is the mutant. But actually, this is the wild type. Why? Why is this the wild type? Why do they call it ebony then?
The well, they have the wild type alleles. I don't know.
So this is the wild type allele. This is the wild type. But it's called ebony. Why? Why do they call it ebony? So genes are named after the phenotype that shows up when that gene is mutated. So if this gene gets mutated somewhere, it will cause the ebony phenotype. So we'll call this gene, we'll call this ebony. Even if a fly has this intact gene, this full gene, it has that intact gene, it will show wild type. But we'll call this gene ebony because if this gene is mutated, the ebony phenotype will show up. So then how do we distinguish this from the ebony thing, like the ebony phenotype or the ebony allele? So the ebony allele will be called E1 or E, this specific name. I think it's E or E1. I think it's E1. So the same goes for singed, for example. If you go to flybase and type singed, what you will get is the wild type singed. Okay, but how do we refer to the singed mutant? It's called SN3, singed 3, SN3. What about vestigial? Well, again, the wild type we call vestigial, and the mutant is VG1, VG3, VG, depending on the type of mutation. So far, throughout the semester, we have been using the word ebony, vestigial, singed, and black to refer to the mutant. And we did that intentionally. We didn't want to introduce this piece earlier in the semester to not to in order not to confuse you, right? So, but now that you guys have studied enough genetics and we're towards the end of the project, so it's time to know. It's like a secret that we've been hiding. So that when we talk about the ebony gene, right, or the ebony allele or the ebony gene, we refer to the wild type. When I say the ebony gene, we're talking about the wild type. If you want to talk about the the mutant allele, you have to say E1 or E2 or a specific mutation. For singed, for example, when you say the singed wild type, the mutant is called SN3, singed 3. Vestigial, there are like numerous versions of the mutant vestigial because the wild type is vestigial. The mutant, there is like VG1, VG2, VG11, numerous vestigial versions. All right, so let me type singed 3. So let me type singe. So this is singe, right? This is the wild type, right? That sequence, if you click somewhere in here, you'll get the wild type. Let's look at alleles right here. So when you see alleles, this is probably too big. Alleles right here. So let's click on alleles. Of the alleles of singe, there are numerous mutations, right? There is the singe 3, that's one mutant. There is singe 68, SAL2. There are numerous mutations, right? Because come on, you have an entire gene, right? Is it only possible that it gets mutated in one specific position and that's it? Well, there are probably multiple mutations. Sometimes this letter is mutated. In another mutant, another position is mutated, right? And that would create multiple versions of the same gene. Is that clear? So when you say singe, although we have been referring to that when we say the word singe or ebony, black, or vestigial, we're thinking of the mutant, right? But when we talk now, when we talk about the wild type, when we say ebony, singe, vestigial, or whatever, that should refer to the wild type sequence. And the mutant, we should refer to that as the name of the allele, singe 3, E1, vestigial 1, V1 or VG2. So I think the same goes for, let's go back to ebony. Here again, we can go to ebony. Ebony, there's EH1, EH3, there is E1, which I think this is the one that you guys have. You guys will have ebony, I think it's E1. So this would be the name of the mutant, right? It's called E1. Same for vestigial, I think it's VG2, I believe, or 3. Okay, so now we know that the sequence we find on flybase is the wild type. And this applies to other species. So for example, if you go to look at the human genome, right? Or if you look at the rice genome, any genome that's published, whatever gene you will find there is generally the wild type, right? The normal thing that is found. They're not going to necessarily sequence some rare mutation. They will first start by sequencing the plain Jane, like human being, right? A general human being, or a normal Drosophila, or a normal mice, or a normal whatever. And once they sequence that, they can look into mutations, right, in their sequence, but the genome sequence of a specific species would be generally the wild type. So now that we've agreed that the ebony sequence, vestigial black or ebony sequences that are on the website, those are the wild type. Okay. So, okay, this is the wild type. Okay, but we want to find the amplicon, which is the specific piece that we have amplified using PCR, because did we amplify the entire gene? We did not, because this is like 7,000 letters, bases. We didn't amplify the entire gene because one, it's too big. We don't really need that. So, and two is we're interested in a very specific region of that gene. So those 7,000 letters, we're not really interested in that. We're interested in a specific area. What is that area and how did we pick it? Why did we pick a smaller section of that gene? Why did we not amplify the entire thing? We mentioned that it's big and we don't need that. We only need a specific piece. What is that specific piece? Yeah, that's where we think the mutation is, right? That's what we want to amplify and look into detail, right? It's like you are watched if there's maybe, God forbid, a crime happened somewhere and there's a footage for that entire week, right? You're not really interested in that entire week. You'll be looking at that specific day at that specific time between maybe 1 and 3 when the crime happened, right? So you're interested in a specific area over time. Okay. So my next question then is, how do we find that specific region that we amplified? How do we find specific regions? How do we amplify that specific region? Primers, right? So the PCR and primers, they identify a specific sequence and they amplify that. You guys remember PCR and how it works? We have two primers, they are flanking that specific region, and they go that way, they amplify the sequence. So a good strategy would be to find the primer sequence and see if they match somewhere in here. So, and if we find that the primer binds somewhere, if we find the location where these two primers bind, then whatever sequence is in between must be what has been amplified. You guys following me? Okay. Let's look at the primer sequences then. So those are the sequences of the primers that you have used. So what we need to find now is like two primers, and then whatever is in between is the piece that has been amplified. Right? We need to get that piece that was amplified to compare it to the mutant, right? Okay, let's find ebony. Let's take, these are the primers that we used, that you guys used this week, last week for the amplification. Let me copy this. Let me go back to this file, and let me do just an easy search. Search, but okay, no match. My primer was not found. That's a problem. Let me find the other problem. Let me find the other primer and see why this is happening. Okay, let's search. Okay, I find this primer. So I found one, which is good. I found this one. But I didn't find this other primer. I didn't find the forward primer. Why is that? We obviously need two primers, so we know exactly what, if we have the beginning, we don't have the end, right? We don't know where it went. So we only found one primer. Why did we not find the second primer? It's in the other direction. Does it bind to the same strand? It binds to the other strand, right? So this sequence is the sequence of one strand, right? This is just one strand. These are not complementary letters. This is just one strand, right? And there's a complementary strand that we didn't provide, right? Because general DNA sequences would provide just one strand, right? Because you could predict the other, right? And it makes it easier. So we're giving you only one strand, right? So the other primer would be binding to the other strand. So I wouldn't be finding it here, right? So if you remember PCR and how it works, if this is a DNA strand, you have a top strand right here, and this is a bottom strand. I don't know if this will work. Okay. These are two strands, right? I have one primer that will bind here and it goes this way. I have another primer right here that goes this way, right? So these two sequences are completely different, right? They are complementary, but they're different. So, so if you search this sequence, you can find this primer, right? But you're not going to find this primer. But if you do a reverse complement this sequence, you can find this piece. If you do a reverse complement of this primer, you will find it here. Because this primer is basically the same as this sequence. So we'll find one primer, but not the other. So the other primer, what we'll do is we'll take this sequence and we'll do a reverse complement, which is, we'll just do DNA reverse complement. Just basically the reverse and as the name suggests, reverse complement. Submit, I get this. So, and I will take that sequence and do a search, put it right here, and now I find it. So this is basically telling you where that primer would bind on the opposite strand. Right, so I can highlight this as well. Now I have two pieces of two binding sites. So I know that this region in between is what is being amplified. Highlight it maybe in something like. So now I know that this region, this is the amplified sequence. This is what was amplified. This is the band you saw, or you would see on the gel. You click here. That's not right. 6,000, that's a lot. Why is it highlighting that type of thing? Okay, let me click here. Okay, 2,380. So that's the piece that was amplified on the gel. If you have singe, it would be 1,200 something. Vestigial, about 800. And black, about 1,500. So now I know that this is my amplicon. So this is the amplicon. This is the amplicon. This is the piece that was amplified. Okay. We now need to compare it to the mutant, since this is the wild type. We've agreed that this is the wild type, right? So we need to compare it to the mutant. But before we do so, so this is the intact wild type. Can we find the protein that this wild type codes for? Can we find the protein? What the protein sequence is? Can we do that? Well, yes. If you go back to here, you would see that this sequence, so the pink sequences, this is the protein coding sequence, the CDS. So all the pink regions are the protein coding sequence, or the exons. Those blue things, blue lowercase letters are the introns, and those uppercase are the untranslated regions. So if I wanted to find the protein coding sequence, I could just eliminate all of those letters and only pick those pink regions, right? I could pink those together. We can do it. So if we wanted to find the coding sequence, should we find the coding sequence within that amplicon, or the coding sequence in the entire G? Well, I don't want you to explain something wrong. So it's the amplicon, but why is it the amplicon? Why is it wrong to find the coding sequence in the amplicon? Why, whenever you're trying to find the protein or coding sequence, you should look at the full gene? Well, because this is the full gene and what will be actually used, like within the plant or within Drosophila, when it tries to produce a protein, well, it will start from the promoter, look at the ATG's first start codon, will translate that. So this amplicon has no meaning biologically. Like the Drosophila doesn't know that you amplified that specific piece. It doesn't even care, right? So that amplicon is something that we artificially designed to look at, right? But when you want to look at the protein, you'd look at the entire sequence, right? You look at the entire gene. So what we can do is we look at this, take those blue letters, pink letters. Let's find, make this bigger. Let's find CDS or the coding region. So I will paste. Let me just remove all the highlighting we have for this. So the coding region, of course, I have to remove those letters. And you can see it starts with an ATG. I would remove this. Remove this. So this is like alternative splicing, or like the spliceosome, or kind of cutting out all the introns. Doing those. And these. And now I have my, okay, there's one extra piece right here. So now I have a coding sequence of how many letters? 2,640. I can take that sequence and just go to type DNA translate XPASI, just to translate the sequence. Translate, and here. This is the protein sequence of my gene. So that's my gene. Yeah, so that's the protein sequence. I can just copy it, take it back, put it here in my file, and this is the protein sequence right here. And your protein is about 800 amino acids. 800 amino acids, 879. So this is the wild type, the functional protein that we call ebony. Okay. Now we want to compare it to the mutant. Now that we know how the wild type looks, let's look at the mutant. For the mutant, let me see if I can. So for the mutant, you will notice that we are giving you two different sequences. We're giving you a sequence called ebony using a reverse primer, and then ebony using a forward primer. And these files won't open. We have to download Finch TV. I don't know if it will allow me to install it on this computer or not. Okay. Never mind. We can still continue our demonstration without that. So we have now the sequence, the amplicon of the wild type, which is 2300. And we're giving you two sequences. For ebony, you'll see that there is an ebony sequence using a reverse primer, and then there's an ebony sequence using a forward primer. The same goes for Singe, the same goes for black, not for vestigial. For vestigial, we're giving you only one sequence to make your life easier. It's just because the sequence didn't work, so we provided you only one file. So But the question is, why do we have two sequences for ebony? Like for the wild type, we just got one sequence from Flybase, which we downloaded and worked with, right? But why do we have two sequences for the mutant? If I have a DNA sequence, if I have a piece of DNA, which is right, if this is my piece of DNA that I'm interested in, so we send that piece of DNA to the lab to get sequenced, right? And we provide them with a forward primer and a reverse primer. And generally, the way sequencing works is that this primer, there will be a tag polymerase, an enzyme that will build this complementary strand. And as it builds it, there is a way for us to know which letter is being added, right? And this is how sequencing works. So you let the primer here, you have the tag polymerase, and that polymerase will build it as if it were a PCR, it's like a PCR. But as those letters are being added, there's a way for the machine to know which letter is being added. It's either a change in the pH or fluorescence, somehow. But there's a way to know which letter is being added. So theoretically, if I sequence only this letter, this strand, I should know that would tell me what the sequence of this strand is. I don't really need to sequence both strands, do I? So because these two are complementary. So, but we actually sequence the two strands. Why? One should be sufficient, right? Theoretically. But why do you have to sequence the two strands? Problem is that as this primer starts and the polymerase starts building, it starts really pretty good, but then the quality towards the end would be really, really, really bad. Right? So the quality of the sequencing would be really bad. So towards the end, we wouldn't really trust what letters are. So we don't really know because for several reasons, the technical reasons, the quality as the polymerase keeps building the sequencing, the quality would be really bad. We would not really trust those sequences. So what we would do then is, since we're good about this part of the sequence, but not this piece, so we'll sequence from here as well. So when you start the sequencing from here, we will be sure about that first part, and then that final part would be bad in quality, right? But we're not worried because we've already got this covered over here. So it's like you have a specific piece of DNA and you want to sequence it from both sides to make sure that you have both ends covered in high quality. Is this clear? So you have a piece of DNA, and because of the sequencing quality, drops as you keep sequencing. So if you start right here, this end of the sequence will be poor quality, meaning if it tells you it's an A, T, C, G, we don't really trust that because it's not really good reads. So we sequence from both ends. So that's why when you have when we give you sequences, you'll have two strands, using a forward primer and using a reverse primer. So, but for the sake of the analysis, let's assume, so this is the wild type. Oh, so this is the wild type. This is the wild type amplicon. So let's compare these two sequences. So this is my wild type. We'll use a website or database called BLAST. And I know maybe you might have heard of it, even if you have not used it. So now we need to compare two sequences. So we'll click Align, two or more sequences, and that will open another window over here, another box. So this is the wild type. And let me just delete a piece in the middle. No, not from this one. Let me just copy that entire thing, put it right here. So this is exactly similar to the wild type. I just copied it, but I'll just delete a piece so we can use it for the demonstration. So when you do the analysis, you'll take the wild type and pick the mutant sequence from the forward primer and do what I'll do. And then you will repeat that process, but you will pick the sequence from the reverse primer, right? So because you have two sequences from the reverse primer and from the forward primer. So, but imagine that this is the wild type, imagine that this is the mutant. Let's just blast, meaning compare. And what we see over here, you can look at the graphic summary. Okay, I think I deleted the most end of it. So let me kind of deleted the vast majority of the sequence. Okay. It should do the I'm just gonna cut a couple of lines over here. So what you'll see is that graphic summary. So the query is what we put on top, which in our case was the wild type, and the bottom is the mutant that we delete the piece of. And you can see that this red line shows you that they go hand in hand together. Then there's a missing piece in the middle that exists in the query, which is your wild type, and doesn't exist over here, right? And then they align again. So from this, looking at this picture, we kind of see that there is a deletion because we just deleted here, right? If you look at the dot plot, you would see that something is broken, something is missing in the middle. So, but the easy thing to do is, since we see that there are two pieces, this piece and this piece matching, and there's a piece missing in the middle, we could go to the actual alignment. And this would show us that there are two pieces. This is one piece, this is another piece. So two pieces of DNA. And to look at this is actually, they're comparing all the sequences line by line. So this is the wild type, this is the mutant, right? From letters 1 through 60. Wild type, mutant, right? And so on and so forth. So this is the first chunk of DNA in which they match. And this is the second of DNA where they match. And whatever is in between is missing, right? So in order to find that missing piece in this file, what I could do is just go find that specific piece, the beginning and end of this piece, right? Find this beginning. I can do it in a second. So I can find this specific sequence, and I'll highlight this right here. So this is my missing piece. I'll maybe highlight it in blue. This is the end of my sequence right here. So this is the end of the first piece. So this, whatever is in between these two pieces, this is the first piece. This is the first piece, which is this piece, right? And I can do the same for the second piece. So this second piece starts with here. So let me highlight it in blue. And the end of that piece is right here. Again, I can search over it. So this is the end of the piece. Are we missing something? Oh, I had an extra space at the end, which is right here. So I can just search for this, highlight it. So this is my second piece. Whatever is in between is what's missing, right? Well, I think we'll leave. Okay, sounds good. So this is the missing piece in the middle, right? And don't worry, over the weekend, once we're done with the writing assignment for these, you can work on that analysis and just do as much as you can, right? We are trying to get you to work with it and struggle a little bit. That's where the learning is, okay? Attendance is up on Canvas. You can do it on your way out. So, and I'll be also outside if you have any questions, you can speak to me. So